Hello and welcome to mini lecture 9 on the scientific procedure. My name is Martin Hughes, and in this mini lecture we're going to continue with the scientific report. Just to remind ourselves, we've looked through all of the 6 steps of the scientific procedure and we're currently on conclusion, so we're writing our scientific reports. In the last video we looked at the introduction and methodology, and in this video we're going to look at results and discussion. So we covered this last week, so if you haven't got this already for your scientific report, please look at the previous video and I navigate you onto the Inspiring Ecology website where you can find the material and methods. As I said, this week we're looking at the results and the discussion. So, by the end of this video, we will have the main bulk of our scientific report completed. In the final video, we will discuss your in-text references, the reference list, and other details that must feature in your scientific report. Just to remind ourselves, we introduced you to the hourglass structure in the previous video, and the introduction we said we wanted to start off with the broadest possible topic and filter that down until we reached our material methods. In the material methods we can't really go beyond the scope of the material methods, and that's the same case with the results. We can only talk about the results in the results section. We don't really interpret the results in that section. We leave that for the discussion. And this is an area that some students kind of struggle with. What should be my results and what shouldn't? So hopefully I make this a little bit clearer through this video. And then finally we talk about the discussion, where you can see the hourglass structure again. We broaden out in the final paragraph of the discussion. We're really tying in our data to real life examples. So we're going to begin at this very narrow point again with the results section. So, like our methodology, we are at the narrowest point in the hourglass. This means we cannot talk about anything other than our results in this section. The results section explicitly states our findings, and importantly, we don't discuss what our findings mean at this stage. We save that for our discussion. And what we would expect to see in the results section, well, we'd expect to see your graphs. We now have a box plot, tables, the ANOVA produced 2 tables for us, and the statistical analysis results. So we have that all in the ANOVA table. So we have all our data handy. So like I say, we're going to have to refer back to our Excel spreadsheets, and we're going to copy and paste our box plot and our tables into our report. And we're also going to have to be really careful about stating the p-value, the f-value, and the f-critical value. The reason we have to be so careful about this and make sure we explicitly state it is because this information will be informative, but also tell the readers, did you reject or fail to reject your null hypotheses? So we really have to have a sentence in there that explains all that. So we're going to look at the data sheet again. So hopefully this looks familiar somewhat. If we open up our data sheet, we hopefully have our box plot in this region here, and you may have your ANOVA on a separate page, which is fine. So simply what we're going to do is open up a new Word document here, and we're going to copy and paste our box plot and our ANOVA and summary table into this Word document. So if I click on my box plot and press Control, copy, open up my Word document, right click again, paste, there we go. As easy as that. We have our box plot in a Word document. Sometimes the formatting goes a bit weird when you put stuff in, but we can mess around with it. At least you know where you've got the original box plot. If I click on ANOVA, it opens up my tables that I have. So this is information that I used in previous videos, and I conducted my ANOVA. So hopefully you have something that looks like this. We're going to copy this summary table into our Word document. So we just Control, copy, into our Word document. And we're going to paste special, and we want to put formatted text. This just kind of helps it a little bit. I'm not too concerned what you do here, but This works best for me because sometimes it goes a bit funny. But if I do something like that, then it puts in a table that I can then increase the size of, and that looks a bit better. And we're going to do the same with the ANOVA table. So just click and drag, highlight all this information, right click again, copy, back into my Word document, and paste special again, formatted text, there we go. Okay, so that's basically what I want to see in my results section. We have the graph, the box plot here. You may want to delete this chart title because it just says chart title. So we can just click on that and delete. We've got our summary table and we have our ANOVA table. I'm just going to close this just now. Okay, so we have information here that's going to be really important for our results. Okay, we have a graph that tells us or illustrates the time data series. A summary table that tells me the count of the data, the average time spent by each of the bird species and the variance, which is important. And the sum total amount of time, that's interesting as well. And then finally we have our ANOVA table. And importantly we have our F value, our P value, and our F critical value. So you can see this hasn't really copied over too well, these numbers, but that's okay. We can fix this later on. But what we really need to do is caption these. We can't just leave them floating around in the results section. So this graph here, in science we would call this a figure. So we're going to call it figure 1 because it's the first figure I have and the only one I have. And you may have more, you may have different charts that you've created, that's awesome. But for our videos we just looked at this. So we're going to have box plot of time series data of wild birds using a feeding station in New South Wales, Australia. Something along those lines. So we need to describe it, we need to have a caption, and for a graph we always have the caption underneath, below the graph. And annoyingly perhaps, the table, it always comes above. So we're going to call this table 1. So it's not a figure, it's a table. Table 1, summary, statistics of time, data of wild birds using a feeding station in New South Wales, Australia. Okay, you get the idea. But we need to describe them, we can't just leave them there. We have to have these captions. The ANOVA now looks a bit rubbish, it's kind of spread over 2 pages, so just click return. And obviously now we have a table 1, so this one is going to be called table 2. And again, I need to give it an appropriate caption, so summary table produced by an analysis of variance test. I'll maybe put ANOVA in there in brackets, just to be sure. On time series data of birds using an artificial feeding station in New South Wales, blah blah blah. Okay, so you can change that around a little bit, but as long as you describe it so the readers understand what they're looking at, we can't make any assumption that they know what we're talking about. So we really need to be explicit and have these captions. And what this allows us to do as well in the discussion is refer back. So for argument's sake, if we have a species, which I think a lot of you have found, there's one particular species, I know I've spelled galah wrong the whole time we've been doing this. If we have another species in here, perhaps, that has a high count data, so it features very heavily in the experiment, and another species doesn't, that's really interesting information. I could talk about that in my discussion and say, and put in brackets, see table 1, or just put table 1. And that allows the reader to go back to your results section and find that information out. So having these captions is really handy to help direct the reader to appropriate information as you bring it up in the discussion. So that's why we have them as well. So once we have that in, that looks pretty good so far. So results section fairly quickly actually is nearly complete for the purposes of this. You may want to expand and talk about things in more detail, but the one thing that we have to be very explicit about is the rejection, in our case, of the null hypotheses. So our F statistic here is 18.04. Our p-value is less than 0.05. It's that kind of weird number, but we know now that that is an extremely small number. And our F critical value is 2.9, which is less than 18. So although we have that information there, which is important, we need to tell the readers explicitly whether or not we rejected or failed to reject our null hypotheses. So I would maybe add another sentence to the bottom of your results and say, based on results from an ANOVA test, in our case, we reject our null hypotheses, which is obviously H0. So we can put H, and how to do the wee 0, if you go up here, there's an X with the subscript. If you press that and do a 0, and then press it again, that's how you do that. So you can have H0. You may want to state your null hypotheses. So I can maybe say that there is no difference in time spent between different species of birds at a feeding station. And then what I want to see in brackets is, well, how did you reject that? So what information? Remember, we looked at the F statistic, the F critical value, and the P value. They all helped us reject or fail to reject our hypotheses. So we're going to, I know it's already here in this table, but we don't want to assume the reader's going to pick that up. We're going to tell them exactly what we found. So F stat, or F statistic, equals 18.04. The F crit equals 2.9. And our p value is less than 0.05. Okay, and you may want to say then, we therefore accept our alternative hypotheses H1 that there is a difference in time spent between bird species at a feeding station. Okay, so I know this might seem a bit overkill and a bit OTT, but we really need to be explicit stating this result here, because this was the hypothesis we set out to test, and in the result section we've categorically said yes, we have rejected this, or no, we failed to reject. Whatever your result was, you would address it accordingly and put it in the same way that I have. So I hope that makes sense. You may have more information you want to talk about, such as the average time spent by birds, the mean time spent, and average time spent, or the cumulative time spent by birds. That's fine. But what I said before in the results, we just state the results objectively. I'm not making any interpretations just now. So although I do have a difference between the birds, I've not started to explain why I think there is a difference, or the magpie spent more time than the galah, for example. We leave that for the discussion. You may want to state that in a sentence and say the average time spent by the magpie was blah blah blah, the average time spent by the pigeon was blah blah blah, and then you go on to discuss it in your discussion. I hope that makes sense. So we're just being very objective here. We're not interpreting the data, we're just stating what we found, and then it's our job to highlight the most important parts of that in the discussion itself. So I hope that's clear. If you can produce something that looks similar to this in your results section, you're doing pretty well. And it's giving the reader lots of information, so we can interpret what you did. And as the reader, I know, okay, 100% they have rejected their null hypothesis based on their data analysis. So that's really interesting. Okay, so close that just now. Back to our PowerPoint. Okay, so we've dealt with the results then. That's the results section complete. You may add more, like I say, but if you can get to that level, I'd be pretty happy with that. And the more you can expand or talk about your results as in stating them, by all means, that's fantastic. And if you have more graphs or more tables that are interesting, please go ahead and include those. I'm not trying to dismiss the results section as being very small, but I don't want to overwhelm you either. For the purposes of this scientific procedure mini lecture video series, this will suffice. Okay, so now we're moving down slightly to the first paragraph of the discussion, which will feature immediately after your results section. So, discussion 1, it's the inverse of the introduction in our last video. In the first paragraph of your discussion, we're at the narrowest part of the hourglass again, in terms of the discussion. So we want to just discuss exactly what it is you found in your results. So what did you find? You may start off the paragraph by saying something along the lines of, in our study we found, or did not find, a difference in time spent at an artificial feeding station between different species of birds in New South Wales, Australia. We found species X spent the most amount of time at the feeding station, and species Y used the feeding station the most. And you may find that one of the species that used the feeding station the most is non-native and not a native species. And that's really, really interesting, because that could lead you on into paragraph 2. Well, is the feeding station good or bad for birds in Australia based on the data you collected, the data that you analysed? So you may start off your second paragraph broadening that out into, how does your data inform us about Australian birds more generally? Did you find anything interesting about the bird behaviour? Can you contribute to the debate about whether or not we should be feeding birds? In my scenario, where I reject the null hypothesis and suggest there's a difference, if I found that the non-native species appeared to be benefiting more from the feeding station than the natives, well, I'd be inclined to say that it's not a good idea based on our data. So we have to be conscious of the fact that our data is limited, and we'll talk about that in the final paragraph. But what do you think? Based on your data, would you recommend feeding birds artificially in Australia? And there's an important topic of debate that I'm going to bring up in a little second. This information is actually going on, or this debate is still going on to this day, and it's very topical actually. So I'll guide you somewhere in a little second. In the third paragraph then, how does your data fit in to the global picture? So this might be quite difficult to think, well, we only looked at data from New South Wales. How can I possibly infer that into the global picture? Well, you can. What is the global narrative for feeding birds? Do most countries in the world encourage it or discourage it? After some research that I've conducted, I'd be inclined to think that most people actually do encourage it. And does your data fit in with the rest of the world's advice or not? So if you found that the non-native species appear to be using the feeding station more, then no, your data does not fit in with the global narrative. And that's really interesting. That's a good talking point. So therefore, that could lead you on to, why is the protection of native birds so important? In our introduction, we talked about pollination and seed dispersal. So if we disrupt the natural flow or the natural populations, or we change the species composition within an ecosystem through artificial feeding, this could have serious knock-on effects, not just in New South Wales, but across the country, and then potentially globally if it happens in lots of different areas. So you can contextualize your own data into the global narrative. I know a lot of my students kind of struggled to connect the dots, but when you explain it like that, you can see, all right, yeah, I didn't realize I could talk about it in those terms. So you can talk about this in really broad terms, and you can see the hourglass again we're broadening out. We want you to tie this in to the real world. The final paragraph then, like we just suggested, that we know that we're just looking at a small set of data. So what were the limitations? The final paragraph would be a good paragraph to remark on the limitations of this study. And there's plenty of limitations. There's no perfect study. And then you could maybe propose future work. So what were the limitations of this study? Well, I've only provided 4 videos, and those 4 videos only occur in one location, and that one location was only filmed at one time of the year. So you may suggest to increase our sample size. If you hark back to the days when we discussed pandas, increasing the sample size allowed us to maybe interpret more from the data, patterns that we couldn't pick up with our limited subsample. So we could increase the sample size. That may give us a more realistic picture of what's happening. I only conducted this in New South Wales, in Brawongal, a very rural area of New South Wales. Why not Queensland, Victoria, South Australia, the Northern Territory? Why not spread that out across the continent and see if this seems to be the case in lots of different locations and different types of locations within those areas? So I was looking in quite a rural, remote area. We might want to look, I think we'd class the area that we looked at as kind of scrubland. Why not more in the bush, or in a desert setting, or in the rainforests in Queensland, on the coast? Okay, we could look at different areas that have different ecological features, different bird populations. Could be really interesting. And we could also not just increase the sample size, but we could maybe do a bit more of a pilot study. I'd be very impressed if anyone had actually completed a survey like this before. And a lot of my students found it quite difficult to identify the species first time around. And once they got their eye in, they became better at it. So it may be useful to increase our ability to identify the species. And one of my students came up with a fantastic idea that perhaps we could construct an arena in front of the camera, and any bird out with that boundary was excluded from the observations. So we only included the bird species that actually walked into this little arena. And that would allow us to dismiss the birds walking around in the background, which were actually quite difficult to identify. So there's a lot of fantastic opportunities to discuss this, and I would encourage you to do so. This not only strengthens your paper, but it demonstrates to me and anyone else who may be marking your work that you fully understand the experimental design, how it could be better, and that you're not extrapolating beyond your data. You understand that this is maybe not a realistic representation of the bird populations generally, but how could we get a better picture of it? So all that stuff would be fantastic. And if you can propose a future study based on this, or based on some research that you've read related to this, please put it in as well. That'd be fantastic to read. Again, that demonstrates another level of ability and your depth and understanding of the subject. So don't be shy. Okay, the one thing I wanted to show you actually before we go ahead was, we were talking about references and information. My fan's being very loud on my Mac, apologies. I was trying to see what the problem was. So, talking about topical issues, this is the current issue of Australian Geographic. I'd like to pretend I had something to do with this, but I really don't. And the first page, front page, says, "Feed the birds, ? Should we or shouldn't we?" So, I know we're looking at a very simple experiment, but the questions we're asking and posing is really relevant and pertinent to things happening in Australia right now. So you guys are contributing to this debate by participating in this study. So if you're watching this in real time, May, June 2018, try and get a copy. If you're watching it sometime in the future, perhaps you can go on the website and look at an archived version. But yeah, just to prove that what we're doing is actually really important. And the last thing I wanted to talk to you about was to navigate you to the website again and look at the online lessons. Something that's going to help you write your report in the practical resources guide is a rough guide for scientific report writing. If you open this PDF, it's a 3-page document. I've broken down how I would approach this with everything we just discussed there. Okay, discussion, results, material methods and introduction, and the abstract. Now, I said we would come back to the abstract, and that's what we're going to do now. I'll talk about the reference list and the appendix in a later video, the next video. So you can download that, I would recommend you do. So just to finish off, we're going to talk about the abstract. Now, the reason I left the abstract till last is because if you've written your intro, material methods, results and discussion, it's already written for you in a sense. So we usually leave writing the abstract last, although it comes first in the scientific report. And like I say, the reason we leave it until the last is because you've already written it when you write the rest of your paper. And the abstract acts as a summary of your paper. It allows readers to quickly gauge whether your paper is relevant for them to read. So our manuscripts may be only a few pages long, they may be longer, but if you have a manuscript that's maybe 25 to 40 pages, you want to, before you go and read everything and try and find your information, you really want to read the abstract, and that gives you a good indication whether or not you should read the rest of the paper. So it's a really useful thing, and I've left it out because, like I say, we've already completed it if you leave it to the end, but I'm not dismissing its importance at all. It's really important. So the best way, or the easiest way to do this, to write your abstract, is take 2 sentences from your intro, 2 from your methodology, 2 from your results, and 2 from your discussion. So that would be a good way to kind of structure your abstract. But we want to be a little bit more sophisticated. You maybe want to paraphrase and make sure it flows. But essentially, if you take 4 sentences, these sentences from their 4 sections, what it's going to tell me as a reader is, what is your paper about? What did you do? What did you find out? And how does that fit into the big picture? Okay, so by reading your summary, I know exactly what your topic is about. So that's your intro. You've told me exactly what you did. What was your study? Material methods. What did you find out? So it's part of your results. We found out there was a difference or no difference. And then your discussions. Why is that important? Well, perhaps we found a non-native species utilised the feeding station more than native species. That is important because it could lead to local extinction of native species, something along those lines. So a good abstract should allow me to tick all those boxes. So the bottom section here, what is the paper about? What did you find? What you did, what did you find? And how does that fit in? Read those questions back to yourself when you read your own abstract. And if you've answered all of those, fantastic, you've got a really good abstract. If you don't answer them quite so well, revisit it and try again. It's an important part. It shouldn't take too long, but you shouldn't rush over it either. I should be able to read your abstract and really figure out what you did in your experiment and what you found out. Okay, so remarkably in 2 videos, we've actually covered abstract, intro, material methods, results, and discussion. So all that leaves is the reference list. I've left that to a video on its own because it's a kind of formatting thing. It's a formality that we need to look at, but the main bulk of the paper is now complete, which is fantastic. And in the next video, I'll talk about reference list and other things that will strengthen your paper. Okay, as always, please like, subscribe and follow. It means a lot knowing that people are following the material. Tell us if it's working for you, what's not working for you. Visit our website and follow us on Twitter at inspiring_eco or myself at Dr Martin Hughes. Thanks very much.